Hey, good afternoon, everyone. Welcome to the Northeast Aquatic Plant Management Society's 2024 Winter Webinar Series, our Spotlight on Species. So I wanted to provide you with more information about the Northeast Aquatic Plant Management Society. Our mission is to assist in the management of aquatic vegetation, to provide for the scientific and educational advancement of members, to encourage scientific research in all facets of aquatic plant management, to promote an exchange of information among members, and to extend and develop an understanding in the discipline. So our members are from academia, government agencies, private consulting firms, herbicide manufacturers, and lake associations. We have annual meetings, annual plant camps, as well as webinars with CEUs, and we offer student scholarships for up to $10,000. So if you'd like to learn more about the Society, I encourage you to visit www.neapms.org. So this session is focusing on water milfoil and water soldier. Before we get started, I just want to go through a little bit of housekeeping. The webinar is set to run from 1 p.m. to 3 p.m. Eastern Standard Time, or actually it should be Eastern Daylight Savings Time, sorry. There will be two 45-minute presentations with a block of 25 minutes at the end of both presentations for discussion and questions for both presenters. So go ahead and please type your questions into the chat box, and the moderator, myself, will read them during the Q&A period. Please note that we were able to offer CEUs for this webinar, so please make sure to check in with Summer Stebbins and answer quiz questions as they arise during the presentations, and I will give reminders as well. The webinar will be recorded and it will be shared via a link on the NEAPMS website. Okay, so our presentations, whoops, let's see. There we go. Okay, sorry about that. So our presentations and authors. So the first presentation will be given by Dr. Ryan Thume of University of Montana. He'll be talking about integrating genetics and herbicide studies to improve water milfoil management outcomes. So Dr. Thume received his Bachelor of Science from Florida State University and his PhD from Dartmouth College. His research program focuses on the ecological genetics of invasive aquatic plants, with specific interest in the role that cryptic genetic diversity plays in invasiveness and herbicide response. He is currently an Associate Research Professor of Genetics at Montana State University's Plant Sciences and Plant Pathology Department. Take it away, Ryan.
Okay, thank you. I'm really honored to be invited to share the work that we've been doing on erosion water milfoil with you. Appreciate the invitation. And let's see. Are you seeing me or my screen or both?
Both. You need to probably switch into slideshow mode in PowerPoint.
Yeah. There we go. So hopefully you're just seeing a slide and no notes and not the second slide and all that kind of stuff now. Yeah, so the work that I'll be talking about is really trying to build genetic tools and resources to provide insight and also assist managers for herbicide applications for erosion water milfoil. So I'll just give you a real quick basic premise of the research program. Sorry, I'm still screwing around with these windows. Anyhow, just interrupt me if anything gets really wonky. I'm trying to move them out of my way so I can see the words that are on the slide. The basic premise of the research is that genetic variation can influence management outcomes, that not all individual plants have the same genes and therefore don't behave and respond in exactly the same way. So our primary goal is to use genetic information to predict herbicide response. And so many of you are probably familiar with things like 23andMe or Ancestry.com, where you can swab your cheek, get a whole bunch of genetic information and learn a lot of things about yourself that you maybe likely didn't know about yourself, that in one way or another is captured in your DNA. And so we're specifically interested in building genetic tools to predict herbicide response to inform herbicide management decisions. The research program includes what I'll refer to sort of both short and long-term approaches. And so I'll kind of break the talk up into those different facets, short and long. So most of you are probably familiar with Eurasian water milfoil, but if you're not, I'll just give a couple of the salient features of the plant for the purposes of the talk. So this is a picture of a plant growing in the lab. It's quite a beautiful plant. It's sort of annoying to many people. Really important biological aspect of this species is it reproduces both asexually and sexually. So sexual reproduction creates unique, genetically unique individuals, just like all of us are genetically unique, save for if any of us are identical twins, then we would have one other person in the world that is exactly genetically identical to us. I'll refer to those clones as strains. And because water milfoil can propagate asexually through vegetative reproduction, those individual strains can be maintained indefinitely within and among water bodies. It also hybridizes with our native species, one of our native water milfoil species, northern water milfoil, which is the sister species to Eurasian water milfoil. In fact, they weren't distinguished from one another until probably 1950s or 60s or so. And this is just a real quick slide. This is just an ordination plot of a bunch of DNA marker data. So each point on this plot is a unique genetic individual. And what you can see is we've got, a bunch of northern water milfoil reference strains. We have two different lineages of Eurasian water milfoil, which we sort of arbitrarily refer to as Eurasian 1 and Eurasian 2. And then both of those have hybridized with northern water milfoil. So at the end of the day, we have a lot of genetic diversity captured in both pure Eurasian water milfoil and hybrid water milfoil. And again, I guess the important part is that these unique genetic strains can clonally propagate indefinitely. What we're really interested in is not just distinguishing different strains of Eurasian water milfoil. What we're most, in our lab, what we're most interested in, and what I would argue probably most managers are interested in, is the variation among strains and how they respond to herbicides. And so that variation is really meaningful to managers, really ecologically meaningful. So that's kind of what we focus on. And then this slide just sort of has, just to illustrate genetic variation for response to herbicides. So we've got some data here for a response to fluridone and some data for response to 2,4-D. Each one of these, on the fluridone side, each one of these boxes or panels is just a unique strain of Eurasian water milfoil, so they're all different from each other. The y-axis is the amount, this was an experiment that we did in the lab, the y-axis is the amount of biomass of plants of each strain at the end of the experiment. The point on the left are untreated plants, the point on the right are treated plants. So that's what every one of these boxes shows. And so this red colored box here is an example of an individual that's resistant, a strain that's resistant to fluridone. So it doesn't decrease in biomass. In fact, it increases in biomass when treated with fluridone. This is 6 parts per billion fluridone, which is a pretty standard treatment in Michigan where most of this work has been done. Here's a strain over here that is sensitive or susceptible to fluridone. And so we see when we add 6 parts per billion fluridone, we get a significant decrease in biomass of those plants. And so in pictures here, right, we've got some plants that this plant, this strain here is over on the right-hand side, treated with 6 parts per billion fluridone, growing perfectly fine. And then the far left-hand side is this green box, this strain treated with 6 parts per billion fluridone and is really hammered pretty hard. So there's quite a bit of genetic variation for fluridone response, and that's something that we're really interested in. Similarly for 2,4-D, this is a dose response curve of 2 different genotypes. So we've got here 2,4-D concentration on the x-axis, and then the percent biomass of untreated plants on the y-axis. And so we see a strain here that sort of has this sort of typical dose response curve, where as we add fluridone, it's on a log scale, by the way, as we add fluridone, we get this kind of negative exponential decrease. Whereas this other strain really doesn't get touched much by 0.5 to 500 to 750 parts per billion 2,4-D. So this is sort of significant and important ecological variation, genetic variation, but this is specifically genetic variation for herbicide response. Okay, so with that background then, the research program objectives in the short term, we're building what we call a catalog of water milfoil strain herbicide response and geographic distribution information, and I'll show you an online application that we have for that. And then in the long term, we want to identify the very specific genes for resistance and use that information to develop genetic tests for those genes. So similar to 23andMe, where you might, from your DNA, find out if you're a carrier for a disease or if you harbor mutations that are associated with the disease. We'd like to do the same kinds of things, but specifically for herbicide resistance. So I'll show you this cool little RShiny app that a graduate student in my lab, Ashley Wolf, and a collaborator at University of Minnesota, Alex Badges, developed over the last year or so. We call it Milfoil Mapper. And basically, it's just an online database of water milfoil strain information. So I'll kind of use it as a way to organize some of what I'll talk to you about today, and I'll give you a quick tour of it. So you can access it through this link, and you can get that through my lab's website, or I can send it to you if you haven't seen it already and you're interested. So what you can do in Milfoil Mapper is you can query the database by geography or strain. So you can pick a state, you can pick a county, you can pick a lake in a county, or by strain. And so you can pick a taxon, that would be Eurasian hybrid water milfoil. Or if you knew what strain you had in your lake and you wanted to find out if we know anything about it, and or if it's present in any other lakes nearby or otherwise, you could filter and query the Milfoil Mapper by the strain. So what I've done here is zoomed in or picked a particular strain. Just the name doesn't really matter, but we'll call it 249. And so when I query by that strain, it gives me a list of all the lakes where this particular strain is found. And so this is a strain that's pretty commonly found in the state of Michigan. And it can print a list if you wanted to print that for some reason. Let's say maybe you wanted to get in touch with people that you know that are working on other lakes so that you could share information about it. The other thing, when you pick on an individual lake, it'll bring up all of the different strains that were present in that lake, and then whether we know anything about them. So this is a little blurry, I apologize, but there's 2 different strains here in this particular lake, one of which we don't know anything about herbicide response, but the other one, which is all these circles on this map, we know that it's sensitive to 2,4-D, and we know that it's resistant to fluorocarbon. And so I would make the argument in the state of Michigan, when you have a lake that's dominated by this particular strain, fluorocarbon is not a very wise choice for herbicide control of that strain in that particular lake. So we feel like this information would be really useful for a manager, right? Knowing what strains are present in the lake and how they respond. And so that kind of sets the stage for what we're doing in the research program. So using Milfoil Mapper as kind of an organizing framework, it's sort of kind of where we want to dump everything that we know about Eurasian water milfoil into this application where people can freely use it and use it to make decisions or strategize or whatnot. So in the short term, what we want to do is characterize more water milfoil strains so that more of the strains that we have in the database have clear information about their herbicide response, and there's some stuff to talk to you about regarding that line of research. And then in the long term, what we'd like to do is integrate genetic tests for resistance. So if we have specific molecular markers that we know are associated with herbicide response, then we can put that information into here and predict whether or not a particular strain would be likely to be resistant or susceptible to a particular herbicide based on some genetic information that we have about it. So again, very similar to what you might have seen if you've done 23andMe or Ancestry.com's version of that. So start by talking about the short-term goal of characterizing water milfoil strains, and I'll talk to you about our biggest bottleneck in doing that is the speed at which and the number of strains that we can characterize in any given time. And so a great focus of work over the last several months, as a year probably plus, has been trying to develop some reliable herbicide response characterization methods in the lab that are more rapid so that we can increase the number of strains that we have herbicide response information for. So here's what a typical herbicide screen setup looks like in the lab. There's maybe not great scale for this, but these are approximately 50-gallon stock tanks, galvanized steel stock tanks. And at any given time, we can probably only work with about 12 of them in the lab. And that's unfortunate because that means there's a small number of experimental units that we have to do an herbicide treatment or an herbicide studied at any given time. So CET just means concentration exposure time. There's a very limited number of those that we can do in any replicated manner at any given point in time in the lab. So what we've been working on is trying to replace these, and it looks like it's working pretty well. It replaced these 50-gallon sort of tanks with 5-gallon buckets. So in the space of, in the footprint of about 2 of these 50-gallon stock tanks, we can fit about 9 of these 5-gallon buckets, which means that at any given time, we can be working with a lot more buckets than we would be able to otherwise. So which decreases the space that we use and also increases decreases the amount of time that it takes to do an experiment, because when we work with these, to get sufficient replication, we have to set this up multiple times to get the numbers that we need. So it takes many, many months. So I'll just show you some preliminary data for an experiment that we did recently that's very promising. This is with 2,4-D, one of the herbicides that we're really commonly working with. So here we've got on the x-axis treatment level, so either untreated plants or plants treated at 500 parts per billion for 3 days, which should be lethal to Eurasian water milfoil, the wild type Eurasian water milfoil. On the y-axis, we have percent biomass of untreated plants. So all the, at 0, all the plants are 100% similar to themselves. We chose these 3 strains that we knew from previous work had different responses to 2,4-D. We knew that this Phelps strain in green was really resistant to 2,4-D, or what we would consider really resistant. This North Arm strain was resistant to 2,4-D, not quite as resistant as Phelps, and then a strain that we knew was susceptible. And these are kind of top to bottom, those plants untreated on the left and treated on the right. So we can see these Phelps really aren't touched very much by 2,4-D. North Arm, not so much either, although there is pretty much a growth effect. And then pretty big biomass effect on an injury effect of 2,4-D on this spring treatment. So we had done this at a larger scale. We've done it now at the 5-gallon bucket scale, and we've seen what we want to see. So those results are really pretty promising. So we think we can implement that approach moving forward. And as a practical matter, the way that we would probably assay for something like 2,4-D is use a 2-step assay. So do a quick assay. This takes about 5 to 6 weeks to do. Just at 1 concentration, that would probably be 500 parts per billion. We'd quickly look to see whether or not a target strain was more similar to the susceptible reference or resistant reference. And then depending upon what we find, if we found something sort of closer to the resistant reference, then we'd probably do a more detailed dose response curve to get more precise information about how that particular strain responds over a range of different 2,4-D concentrations. So that's how we're thinking about moving forward with 2,4-D assays. There's another piece to getting these things done quickly. So one is the space, because if we can do these in smaller spaces, we can do more experimental units at any given period of time. But the other thing that we'd like to do is actually decrease the total amount of time that the experiment is running. And so right now, like a rough estimate to do, to characterize strains, typically about a 14-month process. A lot of that is just the time that well, not a lot. A decent chunk of that is the time that the plants are literally in the buckets being exposed to herbicides. And that's because what we have to do is grow the plants up, treat them, let them grow out for a little while, and then measure them. So we're measuring the actual plant biomass that remains after some period of time. Undoubtedly, there are things going on inside the plant physiologically after it's treated with an herbicide, but before the herbicide has done whatever it's going to do to the plant, that would give some indication of how well that herbicide is working. So we've been testing out this well, it's an instrument, but it's really a method called pulse amplitude modulation fluorometry. So it's sort of a fluorometric way of measuring photosynthetic performance in plants. And so the idea is treat a plant with an herbicide. If it's really getting affected by that herbicide, probably photosynthesis is going to be being affected in a negative way. And that might happen well before we see any visual symptoms, or at least the final visual symptoms that we would see at the end of an experiment. So these are some data from a fluorone experiment, kind of testing that idea that maybe we can pick up on the herbicide effects quicker with the PAM meter than with doing biomass. So each one of these panels is a different strain of water milfoil. They're just named from the lake from whence they came. The x-axis is the concentration of fluoridone in the experiments. This is dose response, anywhere from 0 to 12 parts per billion. And then the y-axis on the left-hand side here, which corresponds to these solid lines, is just the percent biomass of untreated plants. So they all start at 1, and then what we would typically see is a decrease in biomass as you increase fluoridone concentration. The biomass line is the amount of biomass left over for in the experiment after 8 weeks of fluoridone treatment. So many of you might be aware that fluoridone kind of takes a long time, several weeks, to really do its job. It's not a very fast acting herbicide. So these are
Ryan.
Yes.
Sorry, I just need to interrupt for one moment. The folks who are interested in CEU credits, quiz 1 is up in the question box. Sorry, thank you.
Cool. So these are the dose response curves. We have a reference resistant strain here from Townland Lake in Michigan that is not affected by fluoridone really at all. In fact, one might argue that it likes fluoridone, though it probably doesn't really have a taste. But but most of our strains, right, are touched by fluoridone, decreasing as you increase the fluoridone amount. And the y-axis on the left-hand side, on the right-hand side, sorry, which corresponds to these hatch lines, are the PAM reading, so the estimate of photosynthetic capacity 4 weeks after treatment. So in half of the time, halfway through the experiment. And what you can see is that the PAM results halfway through the experiment are tracking the biomass results really well. Again, we've got our known resistant strain really not affecting photosynthesis much at all, whereas our sensitive strains are having that photosynthesis affected a great deal. I'm not going to get into the table here, but this is just to kind of demonstrate that the effects that we would see at the biomass level are also seen at the PAM level. And in fact, I don't have it up here, but you can calculate the percent variation explained in any given statistical experiment. And the percent variation explained 4 weeks after treatment with the PAM meter is actually higher than the percent variation explained with biomass after 8 weeks. So that's a really long-winded way of saying we think we can cut fluoridone experiment time down in half. So between moving to these 5-gallon buckets and having shorter experiments, we should be able to characterize more strains in a given unit of time. So this is really promising, and we're hoping that we can get this implemented in kind of more of an operational way so that we can increase the number of strains we have information for. This, I'm showing you results from fluoridone, and it makes a lot of sense. The reasons I won't get into, but we can talk about later if anybody's interested. The PAM meter probably works really well with fluoridone because of the way that fluoridone's mechanism or mode of action works. And so we're testing it on other herbicides. And the short of it is we expect the PAM to be a good, useful tool for some herbicides and maybe not as good for other herbicides. But for fluoridone, it certainly looks like it's going to be helpful. Okay, so just to kind of think about where this sort of short-term work is moving. The first thing I guess I'll put in a plug, like use it. Use Milfoil Mapper. So that would involve surveying your lakes. So we get strain identifications for those. And then if you have strains that you're coming across that you're genuinely concerned about, I mean, like, I would, by that I mean something like, an herbicide treatment was done and it really didn't do, it's very clear that it didn't do what was expected of it. Then we would want to get a hold of that strain so that we can characterize it, have some hard data that we can share with people, and then those data could be applied to any of the other lakes that contain that strain. And it's fairly common that we see strains occurring in multiple lakes. A couple of other things with it, scaling this, it has a little bit of challenges. I mean, we're one lab. We have we have the sort of personnel and space that we have. We're certainly eager to collaborate with other folks on identifying strains and characterizing them. So it'd be cool to have a set of protocols that multiple labs could use, to characterize strains. And, obviously, 2 labs can do twice the work that one can do, and so on and so forth. Regarding funding for this, historically it's been funded by a variety of different things. We initially started with the state of Michigan, who very generously is given quite a bit of funding a number of years ago that really kind of got this whole thing kicked off. Probably the lion's share of funding has come from the Army Corps, U.S. Army Corps of Engineers Research and Development Center, and then we've had some dedicated funding for the development of milfoil mappers, specifically from the Great Lakes Restoration Initiative, which is part of the Fish and Wildlife Service. Currently, that funding is all gone, and so it's currently essentially a fee for service. So when people send plants over, we just invoice and charge for whatever it is that we're doing. I'd certainly be keen to talk to people if they knew of clever ways or funding opportunities to keep this work going, if that seems of interest to folks. Okay, so that kind of wraps up what we're trying to do in the short term. So the short term would just be keep building up this catalog of water milfoil strains. In the long term, it'd be more efficient, I would argue, or at least in my opinion, to integrate genetic tests for resistance, which involves these two steps. One, doing what we call genetic mapping for resistance, and I'll show you how we approach that in the next good chunk of slides. And then developing molecular markers that can assay whether or not an individual strain is resistant or not. So let me take a minute to just try to explain why this is really important. So back to our Floridon data over here. So these are just 8 different strains of Eurasian water milfoil, and this is their response to Floridon untreated plants versus treated plants. And with our DNA fingerprinting that we use to distinguish different strains, if we then characterize those strains in the lab, then we can distinguish strains that have different responses. So for example, these 4 strains on the bottom here are all susceptible, what we would call susceptible to 6 parts per billion Floridon. And then these strains at the top are what we would consider resistant to Floridon. And so we can sort of exhaustively go through each strain of water milfoil that we identify and characterize its Floridon response, and just have a really large catalog with hundreds of strains of Eurasian water milfoil having responded to Floridon. And that's useful, and that would be really useful. But there's a problem with it, a number of problems with it, and probably the biggest problem is that water milfoil can reproduce sexually, and so new strains are being produced all the time. So it's not like the god of water milfoil made 100 strains and said, here you go, here's the 100, and that's all that there's ever going to be. They're being made all the time through sexual reproduction. And what's important about that is a strain like this, a resistant strain, if it engages in sexual reproduction, either by selfing with itself or outcrossing to another strain, it then passes on those resistant genes to the offspring from that mating. And there's no way with the methods that we currently use to detect that a particular individual came from another individual that is resistant, right? There's just no practical, logistical way to do that. And so that means for every new strain that we discover, we have to go through this process of characterizing it. And that's pretty cumbersome when all is said and done. However, if we identify the specific genes for resistance, then each new strain that we identify, we can just ask it genetically, do you have this resistance gene or do you not have this resistance gene? And I'm sort of oversimplifying this in some ways, but that's capturing the salient feature of what we would be going for in a genetic test. So, this is really common in terrestrial agricultural weed management. It's very common for people to collect plants in a field for doing an herbicide treatment and testing for known molecular markers that predict resistance or not. And so this is a figure from a review paper that was sort of featured in a scientific journal several years back. And the basic premise of it really is that it's a lot like your COVID-19 tests, right? So you can do a home kit where you get this kind of colorimetric test of being positive or negative. There's also sort of, you probably all have heard about this sort of PCR test, right? The PCR test is probably a little more accurate than the home testing kit, but the point is that there's various ways to assay. If you have the appropriate molecular markers in biotechnology, there's various ways to assay for the presence or absence of something that we're interested in. In this case, it would be assaying for the presence or absence of a known resistance gene in any individual plant. So that's what we'd like to build. And this is not, we really can't claim that this is anything really unique to us. This is very common in a lot of walks of life, and in particular in terrestrial agricultural weed management. I'll give you an example from aquatic plant management, just to kind of hit it home in that space. So many of you are probably familiar with hydrilla. So this is hydrilla verticillata. This is dioecious hydrilla here. These are dose response curves for 4 different biotypes. We've got growth on the y-axis and fluoridone concentration on the x-axis. These are 4 different biotypes that can be distinguished in how they respond to fluoridone. So the wild type dioecious hydrilla gets hit pretty hard at 12 parts per billion, but we have these other 3 biotypes that are not hit nearly as hard by fluoridone. We have a very resistant fluoridone resistant strain that, you need to get up to 60, 70 parts per billion before you're really having appreciable impact on that particular strain. And so the ability to use fluoridone against that strain is pretty limited in a lot of water bodies. In the case of fluoridone resistance for hydrilla, we know very precisely the exact DNA mutations that confer these different levels of resistance. And so we can very easily just sequence this small piece of DNA, find out what particular nucleotide occurs at these 3 positions, and know whether or not it's this strain, this strain, this strain, or this strain. So basically, we want to do the same thing. We want to find the mutations in water milfoil that determine resistance versus susceptibility, and develop tests for those. So how do we do that? I'll walk you through kind of the sort of genetic and genomic methodologies that we use for that. So the first thing is we have to create what are called genetic mapping populations. And so the way that we do that is we cross a known resistant strain, let's say this one here, with a known susceptible strain. When we do that in the laboratory, we can breed this plant with this plant. And when we do that, we now have a population of brothers and sisters, if you will, that all have, we know that the genes for resistance and the genes for susceptibility are in that collection of individuals. And then we can test to see whether or not those brothers and sisters, say, segregate for susceptibility versus resistance. So some of you you might have blue eyes, but you might have a sibling with brown eyes, or vice versa, right? So that's called segregation, when 2 related individuals show different traits. And so here we've got the results from a cross between this susceptible parent, which is over here, and the resistant parent over here. And then these are just 4 representative individuals that are progeny or offspring from that cross. So we have some progeny that resemble more the resistant parent and others that resemble more the susceptible parent. That's just a snapshot to illustrate it visually. These are the data for those. So the red boxes are the resistant parent. These are resistant parent untreated. And this is how well they grew. These are the resistant parent treated. So you can see that treatment really doesn't do much at all to this strain. This is the susceptible parent down here treated. And so it really gets hammered pretty hard. It looks like this. And then we have this kind of continuous distribution of offspring that range from being pretty crappy growers when treated, like their susceptible parent, all the way to pretty resistant individuals that are resembling more the resistant parent. So what we can do then is we can selectively, sequence the entire genome for some number of individuals that are exhibiting more resistance to fluoridone, and compare that to individuals that are a collection of individuals that are showing more phenotypes that are susceptible to the herbicides. And these numbers aren't exact. We did about 20 to 25 of what I'll call the resistant bulk up here and the susceptible bulk down here. And this is work done in collaboration with these sort of, genomics wizards at the Hudson Alpha Institute. This is Alex Harkis and Josh Clevenger, who are collaborators on that. This part of the research program. I'll skip over this. Well, no, I won't. So we also did this for a second resistant strain. So we've identified 2 distinct fluoridone resistant strains. And so we went through that same process with this second strain. And what that allows us to do is ask whether or not we see the same resistance mechanisms or different resistance mechanisms. And as a practical matter, the reason this is important is in the results slides that I'll show you the results side by side for both of the 2 resistant strains that we have gone through this process with. Before I get to the results, I'll just really quickly try to give you the essence of how this analysis works so that I can, so you have a little bit of a foundation for the graphs and figures that we're looking at, because they're a little funky. So the rationale is that each plant, right, is genetically different. No two plants in these mapping populations are identical. There are no identical twins. Every individual plant is distinct. They only got their alleles from the same parents. It's just the combination of alleles that they got from their parents that makes them different from one another. And so what that means is that if I were to randomly grab, let's say, 10 plants and put them in a group, and randomly grab another 10 plants and put them in a group, and then statistically compare those two groups, on average, any two randomly chosen groups of plants would be genetically similar overall. So each individual is different, but the collection of individuals, if I randomly assign them to two groups, would be very genetically similar to one another, statistically. So on average, these groups won't be different, except for if the way I put plants into groups is to say, well, I treated you with fluridone and you were either resistant or you were susceptible. If we do that and separate that into two groups, then what we'll have is that genetically across the genome, anywhere that's related to, any spot in the genome that's related to resistance versus susceptibility is going to be different between those two groups, because that's how we segregated the two groups. But everywhere else in the genome that's not related to resistance or susceptibility, everywhere else on average, those two groups are going to be very similar to one another. I'm simplifying a little bit, but that's the basic rationale. So that means that what we can do is we can go across the whole genome and find every spot in the genome where there's any genetic variation at all, and look to see how different the resistant bulk is from the susceptible bulk. The way that might work, let's say we're cruising along a chromosome here, and so left to right, we've got these spots on the chromosome where there's some genetic variation. Let's imagine we compare the percent for the resistant versus the susceptible bulk at each one of these spots across the genome. These are contrived hypothetical data just to illustrate this, but so at the first spot, 50% of the individuals in the resistant bulk had a T, and about 50% in the susceptible bulk had a T. So there's no difference between those two bulks. Same for the second spot, 25% have a G, roughly 25% have a G in both groups. So we would say that those particular mutations or that genetic variation is not associated with herbicide response. But if we came to a nucleotide where there was a big difference in the proportion of individuals that had them in the resistant versus susceptible bulk, then that would be associated with herbicide response. So in this case, I've made an extreme, but we say 94% of the sequence reads in the resistant bulk have a T, whereas only 8% have a T in the susceptible bulk. Now we've got a statistical difference between those two groups, and we would say this is a part of the genome that's associated with herbicide response. So that's a little bit of background for how we'll be looking at these graphs in a little bit. We're always going to be looking at the difference between the resistant bulk and the susceptible bulk. Okay, so we do all this genome sequencing, and so let me just give you a quick orientation to what the Eurasian watermuffler genome looks like in terms of how it's what its structure and composition is. So it's about 780 million nucleotides long. That's actually a pretty small genome, and that's the haploid version. So that means each individual receives 780 million nucleotides from its biological mother, and 780 million nucleotides from its biological father. So there's 21 pairs of chromosomes. Each chromosome, you get one chromosome from mom, one chromosome from dad. Those 21 chromosomes are just listed here, and this will come up in subsequent graphs as well, which I'm kind of showing it to you now. So these are just the 21 chromosomes listed top to bottom, and then the length of this bar, it's really not that relevant, but just to have something visually for you up there, is the length of base pair, relative length and base pair of that individual chromosome. It's an allohexaploid, which is a little bit of a pain in the butt, which means that these 21 pairs of chromosomes have some structuring to them. Really what they are is 7 sets of 3 very similar chromosomes. And the terminology homologous doesn't probably really matter for the purposes of this talk, but so in other words, these chromosomes are grouped into sets of 3. And we'll be looking at graphs that kind of do that. So these first 3 chromosomes, they're different individual physical chromosomes, but they share the same genes and roughly in the same gene order as each other. And the same thing, 4 to 6 are have similar genes and are structurally very similar to one. And so what I'll walk through is sort of the results from some preliminary work that we've been doing on this, and that leads to identifying some areas of chromosomes that we're interested in looking at further because they look like spots that are associated with fluoridone resistance in this case. So for these next few slides, I'll be highlighting the set of homologous chromosomes that we're looking at, and then within a column, top to bottom, it's just those 3 chromosomes. So this would be chromosome 1, 2, and 3. And then we have our Lansing mapping population. That's one of the fluoridone resistant strains, and our town line fluoridone resistant strain. And we're just basically looking to see if they look similar or not. So what each one of these individual graphs is from left to right is sliding along that particular chromosome. So sliding along chromosome 1 for the Lansing population from the very first nucleotide all the way out to the last nucleotide. So this is about 45 million nucleotides long here. The y-axis is just the difference in the frequency of a particular nucleotide base pair or base for the resistant population for the susceptible population. You can't really see them probably, but maybe if you squint your eyes really quick, you can see a bunch of gray dots on here. So those are the individual data points. There's many, many thousands of data points on each one of these graphs. And that's just the difference at that particular spot in the genome, resistant versus susceptible. And then the blue line is the rolling average of that. And so it's just a way to kind of take all of those dots and make it into something a little bit more interpretable. And what we're looking for in these graphs are peaks. So spots along the chromosome where the resistant population is unusually different from the susceptible population. And so for chromosomes 1 through 3, we see we're seeing kind of what's looking like a peak on the far left end of this chromosome. And so this is a gene area that we would be interested in looking more at to explore what might be going on for resistance. Whereas these long stretches where the line is pretty flat are not particularly, there's no evidence that there's anything really going on with resistance there. And then just for fun, well, not just for fun, but comparing the town line population, the statistical power for the town line population is much lower, so these are going to be a little more jagged. But we see something similar. If you squint your eyes, it looks like there's some peaks maybe going on in around the same region in the town line population. So if we're prioritizing regions to explore, we're going to find those that have the biggest peaks, and we're going to look for peaks that are present in both our Lansing and town line population. So that's an example of a spot that we'd be curious about. We don't see anything interesting on chromosomes 4 through 9, so I'll skip over them. We do see some peaking on chromosomes 10 through 12, so we see a nice peak here that looks pretty promising. And you can ignore this red line for a minute. If I had more time, I would talk about that. But here's another area where it looks like we have there might be something going on, particularly when we see something like this that's really clean and clear. That's a real nice signal. And then we skip over 13 to 15 because we don't see anything interesting. We see another spot on chromosomes 16 to 18, so another sort of looking like we're seeing some peaking. And then another doesn't even look like it, but this is actually our top hit. This is the one gene region that looks like it has the most highest association with fluoridone resistance in this population. So hopefully that gives you a sense of how we go hunting for these genes. Okay, so looking out into the future for this, this is where we're at with it. I think we have this genetic mapping workflow is down. We have the basic workflow down, but through things that I won't get into the gory details of, we're working on refining our methods so that we can increase the likelihood of finding those particular mutations that cause resistance. And this is a very short laundry list of things that we're kind of working on to improve, to kind of make those peaks jump out a little higher and also a bit narrower so that we can hone in on places a little more easily. I think in the interest of time, I'll save trying to like really explain any of that. Then the next step, once we find spots, is to develop and test molecular markers that are associated with resistance. So I'll give you a couple minutes of how we do that. So ideally, we'd identify the causal mutation for resistance, like that DNA change that is the actual mechanism of herbicide resistance. So going back to our fluoridone, hydrilla fluoridone resistance, we know that already. We know the exact mutations that lead to resistance and how much resistance those mutations confer. That's the gold standard. That's what we're really looking for. We don't have to do that. As long as we find markers that are closely, that are linked to the herbicide response, then they can be a good enough asset. So the way this works, we see this peak here. That's because there's a whole lot of individual nucleotides that have large differences between susceptible and resistant. And so one of these is likely like the mutation that causes resistance, but there's thousands of them here that are elevated in terms of being different between resistant and susceptible. And so in theory, any one of these ones could be good enough, even if it's not the mutation, it could be good enough as a predictor for resistance or not. Another nuance to all of this is that these molecular markers, when we begin building them, can be either DNA-based or RNA-based. And so in some cases, the mechanism of resistance may be due to gene expression and not necessarily just due to, well, it might be due to gene expression. And so I'm not going to go into this. We can talk about it later if anybody's interested, but we've been doing a lot of work on comparing gene expression between resistant and susceptible strains as a way to start finding out if there are predictable patterns of gene expression differences, then we could build molecular markers based on gene expression as well. And so these are two strains, a resistant strain and a susceptible strain. The susceptible strain is on the left, the resistant strain is on the right, and all you really need to get from any of this is that the color patterning on the left-hand side tends to be different from the right-hand side. And so that means that the expression of these genes is higher or lower for the susceptible versus the resistant. And that's something that we can use to our advantage, try to develop molecular markers for resistance. And then finally, when we have molecular markers that we think will work for resistance, we need to test them on uncharacterized strains. So all this work is being done on experimental populations that we've made ourselves to find these strains. What we really need to know is, do they actually work? Do they actually predict resistance or susceptibility when we go outside of our individual mapping populations? And that's going to be a function of how many different ways there are to be resistant. So if there's really only one way to be resistant to an herbicide, then that's going to apply broadly across all the different strains that we see out in nature. If there's lots of different ways to part in the expression, but proverbially skin the cat, then it might mean that any individual molecular marker translates very narrowly to that portfolio of strains out in the real world. And so this is kind of all the, I put this in the long term because this is kind of all the hard work that we need to get into. I'll save the short-term and long-term benefits of relevance. Hopefully I've convinced you that having this catalog of resistant strains versus susceptible strains could be really helpful, and then identifying the genetic basis of resistance could be really helpful. The other point that I'll make is that we hope that this is what we're doing with this in this particular system will be maybe inspirational or practically provide conceptual and methodological models and tools for looking at this in other species as well. I think this stuff will probably apply as we pay more attention to genetic variation of aquatic plants. I think we'll see that this applies more broadly than just your water method. I'm going to thank a bunch of people because there's a lot of work that I got into, and I'm kind of rushing because I feel like I'm probably at my time limit here. But these are 3 people, current members of the lab, that have done a whole lot. Greg Clark's a postdoc who's done a lot of the bioinformatics work. Ashley Wolf developed the milfoil mapper application, is doing a lot of the susceptibility screening. Del Haney has a big hand in a lot of this as well. These are sort of former and past members of the lab that have been really helpful. I already mentioned these collaborators. This is Ray Newman at the University of Minnesota, who's been really an instrumental part of a lot of this work, and then it's been funded by a variety of different sources, most probably the lion's share being from the U.S. Army Corps of Engineers. With that, I'll stop. I don't know if there's time for questions. I suspect not, but I think there's time later on. Thank you for
Yes.
listening. Appreciate it.
Thanks, Ryan. Okay. So I can introduce our next speaker. Hoping I can share the screen. Maybe? Okay. Sorry, folks. I'm having some challenges with the slides. All right, I can't seem to get to the move, so I'll just introduce and read. So the next talk is by Robert McGowan from Ontario Federation of Anglers and Hunters, talking about a decade of managing water soldier in Ontario. Robert's passion for the outdoors has led him to a career in the environmental field. For the past 10 years, Robert has worked as the aquatic project specialist at the Ontario Federation of Anglers and Hunters, where he is working with the Water Soldier Working Group to monitor and control invasive water soldier populations on the Trent Severn Waterway, Red Horse Lake, Black River, and private ponds across Ontario. Previous to that, Robert gained valuable experience while employed with the Ministry of Natural Resources and Forestry, Credit Valley Conservation, Trent University, and Fleming College. Thanks, Robert. Take it away.
Thank you. Thanks for having me, everybody. My name is Robert McGowan. I'm the project specialist at the Invading Species Awareness Program. And today I'll be speaking to you about water soldier in Ontario waters and the 10 years we've been trying to eradicate / manage it and the methods that we're doing to carry that out. So a quick overview, I'll give you a quick background on what water soldier is. We'll look at water soldier in Trent Severn Waterway, where it's ground zero, where it was first detected in North America. The Bay of Quinte, Trent Severn Waterway is attached to Lake Ontario, which the Trent Severn Waterway flows into the Bay of Quinte. The Black River, Red Horse Lake, and what we've been doing about water soldier in private ponds. So a bit about the Invading Species Awareness Program. In 1992, the Ontario Federation of Anglers and Hunters partnered with the Ontario Ministry of Natural Resources and Forestry to establish the Ontario Invading Species Awareness Program. And this was to address the increasing threats posed by invasive species in Ontario. And our objectives are to generate public education and awareness of aquatic and terrestrial species and address key pathways that contribute to the invading species introductions and spread, and facilitate monitoring, tracking of these new invaders that we're finding. And since then, we've taken on some management roles as well. So we also have the mystery sail management program. We have the water, so we're highly invested in the water soldier. Sorry.
Pardon me for one moment. Sorry, we've got our quiz number 2 for CEUs.
Okay.
Thank you.
We're heavily involved in the water soldier management program and several other wild pig programs, etc. So let's just get into what water soldier is. So water soldier, it's a floating aquatic plant native to Europe and Northwest Asia. It was first detected in the Trent Severn Waterway. This was the first detection in North America in 2008. It's been identified by the Great Lakes. Sorry, I'm trying to move some things around here. It's been identified by the Great Lakes governors and premiers on their least wanted list in 2013. It's been listed as a prohibited invasive species in Ontario. So as of 2015, the Invasive Species Act was implemented, and water soldier is listed as prohibited. So that means you can't buy, sell, trade, possess, or transport it within the province. And eradication and management are led by the Water Soldier Working Group, which is a collaborative between federal, provincial, government, non-government organizations, academia, and conservation authorities across Ontario. So why is water soldier a problem? So water soldier forms dense mats of floatings of submerged and emergent vegetation, and it crowds out all of our native vegetation, and this causes decrease in biodiversity. These dense mats of floating water soldier can hinder recreational activities such as boating, angling, swimming, hunting, etc. And sharp serrated leaf edges can cut anybody who handles a plant. You can see it in the picture here on the left, those sharp serrated margin of spines there. So water soldier has two forms. So there's the submergent and the emergent. The submergent form lays in the substrate of the water body, and it is flaccid leaves that are brittle and spirally, and they're very bright green and sometimes have a darker purple color towards the center. When this plant, when the temperatures in the summer start to rise, some of these plants, if it's in the correct water depth, start to photosynthesize, and those outer leaves become stiff and erect. So they fill with photosynthetic gas, which helps them become buoyant, and they float to the water surface. We usually see emergent plants anywhere from 0.5 meters to 1.5 meters. Any deeper, just the plant doesn't seem to be able to breach the water surface, and it stays as submergent form. And both of these forms have offsets and turions. So it just reproduces asexually. So you can see in the bottom right-hand corner, that's an offset. So that's like a baby plant attached to the mother through a runner. And each of these mother plants can have several of these offsets. And in one growing season, these offsets can grow into a mature plant and produce its own offsets. So it can be exponential growth. And these turions and offsets up here are vegetative buds that can fall off the mother plant and sprout into its own little plant and start to grow. So the ecology, so in the fall, the outer leaves that in the summer became filled with photosynthetic gas and became buoyant, they start to die off, and you can see the browning on the leaves on these pictures. So they become waterlogged, and this is what helps sink the water soldier plant to the bottom of the substrate. And that's where it stays there and overwinters in a rootless state. But you can also see how green that rosette is. So that rosette does not go dormant. It is an evergreen plant, so it is alive all summer, all winter long. Here in Ontario, we usually have ice cover, so under the ice, and it just forms a function where it stores its starch in that rosette, and it never really goes dormant. So what happens in the spring when the ice is off and the sun comes up and the water temperatures rise and the plant starts to photosynthesize, it has a really competitive advantage over our native species that don't get that initial jump. So this is one of the reasons why it's become such a problem. So the Trent-Severn Waterway was the first detection of a water soldier in North America. The Trent-Severn Waterway is a canal that connects Lake Ontario to Lake Huron, and it's about 386 kilometers long, or 240 miles. It has 44 locks and multiple hydrological dams that kind of break it up. So I say canal, in portions of it is like a canal, but in others, it kind of has been flooded and it almost becomes lake-like. And that's where most of these plants are falling out, in these larger areas in the river. And as you can see, about 50 from Hastings right here down to the Bay of Quinte, which is part of Lake Ontario, is the main infestation. So that's about 50 kilometers or 31 miles. And this is just a great little timeline series. Without control, water soldier will spread. So you can see in the summer of 2013, along the shoreline here, you can see some plants here, a few sporadic plants. But then in summer 2014, you can see that it's kind of filled that whole littoral zone in, and it's getting very dense. These are pictures from a landowner, and she kind of did this timeline series. She had to change her perspective here to kind of demonstrate how this plant is moving into this bay here. And by September 2015, it's covered the majority of the bay. I know you see some open spots here, but those are more than likely some of the spots where the plant hadn't become emergent yet. It's in its submergent state. So with that said, in 2008 to 2012, we were managing the plant we weren't very organized. We didn't have a working group. So in 2013, the working group was formed. An integrated management plan was developed. And so this is just to guide our monitoring and management and give us some tools in our toolbox to, deal with this plant in some coordinated fashion. So the management objectives are to prevent the introduction and spread of water soldier to new locations in Ontario, implement early detection and rapid response actions to locate and eradicate new water soldier populations, and eradicate established water soldier populations from public waters, and work with the private sector to identify and eradicate water soldier from private waters in Ontario. So our control options, we have utilized herbicide every year. We have an annual management program where in the fall we deliver herbicide to these populations in the Trent Summer Waterway. We conduct manual removal. We have not explored diverse hits of suction harvesting yet. Most of the areas, as I mentioned before, where water soldier persists is that littoral zone between 0.5 meters and 1.5 meters. I'm still interested in investigating it, but some of the areas really aren't conducive to the diver assisted suction harvesting. I'll get into that later. It might be better suited for Bay of Quinte. We've utilized bottom barriers or shade cloth enclosures, which have been really successful, but have their limitations. And mechanical harvesting. We have done a few mechanical harvesting trials, which I'll get into shortly. So the Trent Severn Waterway Monitoring Protocol. So every year in the summer, we go out and we have our lake areas gridded with a 50 by 50 meter grid system, which every point intercept has a unique identifier. And we go there and we can visually check. In the Trent Severn Waterway, very easy to visual check, but we still do a rake throw or rake drag because it's difficult to see sometimes. You can get into some turbid areas. And we have a scale where water soldier scale where 0 to 3, so 0 being no water soldier present, 1 being a single plant, 2 being scattered plants, and 3 being a dense colony. And from this, we also record the top 3 other plants that are present at the point. So this gives us a good knowing where the water soldier is and how much there is, more qualitative than quantitative data, and gives us good baseline for plant community and see how the community is recovering after we do our herbicide treatments. And we also record water depth, obviously, because we are going to be doing herbicide treatments, and that information is pretty important. So on the Trent Severn Waterway, historically, we've been using diquat dibromide, label name reward. We hire a company from the States to come up. They have 2 airboats with crew and a skiff. Usually we spray in October for a few reasons. One assumption is that most of our native plants have gone dormant. And up here, Trent Severn Waterway is in cottage country, and a lot of the cottagers have gone home. So we're not interrupting any of their activities during prime time summer. We do a surface and subsurface treatment, and not all areas get treated. With the Trent Severn Waterway being a flooded waterway, there are a lot of areas that are very inaccessible. So the airboats or even a john boat would have difficulty reaching these areas, and we have trouble reaching these areas to even monitor. So we'll get into a bit of that. So we can see here, this was the 2023 treatment map, and you can see up here, we call this part of the river Lake Seymour, down through Crow Bay into Percy's Boom and Wilson Island. Some of these areas down here in Wilson Island are very difficult to access, and we are not able to treat them with the herbicide. In 2023, we treated 148.9 hectares of the waterway, just the Trent Severn waterway. We have seen success. So there's been a reduction in the abundance through our initiatives through time. So you can see the top map is 2015. We're sitting at 162.55 hectares. And in 2019, we're down to 23 hectares or so. That's been a reduction of about 85% in some areas. And the public sees improvement, which is good. A lot of these cottagers were not very happy that you saw that timeline picture. It virtually made their waterfront unusable. So we have seen great results with the herbicide treatments, but there are some issues, and it goes to those areas that we can access, one, and two, this is a flowing system. So there is a channel that flows all the way through the system, especially in these larger lake-like portions. And we're noticing that water soldier that is adjacent to this water flowing is a little more difficult to address. So this persistent map is made up over the past 5 or 6 years. And these little areas here just keep popping up, and we're trying to figure out how we're going to address these problem areas that aren't as susceptible to the herbicide treatment. And we've had to adjust some of our monitoring to that as well through the use of drones and underwater cameras. So in 2023, the federal government, Parks Canada, they held a pilot control project. So this is one of those areas that is very inaccessible and could, pose as a source population to just keep feeding the waterway with an unlimited amount of water soldier. So we hired a company to come in with the Trexar and manually remove these populations. We've kind of gone back and taken a look, but we're really gonna have to wait to the spring of 2024 to get in there and assess the situation. We've taken the biomass out. It should just be us going back in and picking up the crumbs. The majority of the water soldier came out and was dumped onto dry land. And again, those persistent areas along the sides of the water flowing, the channel, we have gone out and we've done manual removal. We've seen this one spot we're in particular right here. It's been treated every year since 2014, and the population has not been reduced by any. So we've gone out and we've meticulously hand removed them with rakes and packed them up, and we'll have to see what our results are in the coming year. So moving on, the Trent Severn waterway, we've been managing it for I guess 10 years now, with the hopes of eradication. We're planning on staying adaptive and trying new management approaches to reach that eradication status. But unfortunately, with a floating plant and a flowing system, water soldier did find its way into the Bay of Quinte, which is part of Lake Ontario. So in 2021, in September, we found water soldier near CFB Trenton, and being so late in the year, we weren't really able to address the situation. But in 2022, we came up with a tiered approach with eDNA sampling, surveillance, vegetation surveys, and follow-up investigations. And we did the same in 2023. So the Bay of Quinte, Quinte Conservation, which is a conservation authority, here in Ontario has a water monitoring and coastal wetland surveillance program where they go out and they take these water samples and vegetation surveys once a month in the summer. And so they were so kind to tack on the eDNA collection for us during those months to kind of give us a sense of where is Water Soldier, right? So it's a pretty big area, the Bay of Quinte, which does flow into a larger area, Lake Ontario. So we're have a couple of hurdles in terms of manpower and how much can we like do traditional monitoring on. It would take a very long time. So we're using eDNA to kind of direct our efforts. So Quinte Conservation goes out and they take water samples and they run it through the syringe method of eDNA. So it's pretty grassroots method of taking eDNA. It's a syringe and there's a filter that attaches to the bottom of the syringe, and you fill the syringe with water and you push water through. You push as much water through until it becomes too difficult to do so. And then these samples are sent off to the lab, and if there's any hits on Water Soldier eDNA, that's when we go out and kind of direct our efforts to investigating if Water Soldier's in the area. So eDNA is a fairly new technique. It's emerging. So when we get the results, we have to take it with a grain of salt. It's been explained to me, it's like a smoke detector. If your smoke detector is going off, is it your toaster that's making it go off, or is your house on fire? That's a pretty good analogy. It warrants investigation. And this is how we're using eDNA to kind of manage the resources we have. So you can see here, we've had some, the red dots are positive eDNA hits. So that we would go out and we would overlay our 50 by 50 meter grid with our unique point intercept identifiers, and we'd search for the Water Soldier plants there. And it's been fairly successful. We have gone and done this and not found Water Soldier, but we're doing our due diligence by looking. And so when we go out and do this, we follow the Water Soldier monitoring protocol with a 50 by 50 meter grid, the top 3 species, the depth, what substrate there is, and we do the rake tosses. We've also implemented underwater cameras. The Bay of Quinte is large open water. So where the Trent Severn Waterway is a shallow eutrophic environment, we're starting to get into a bit deeper water. It's not as easy to find individual plants. The water turbidity is different. The hydrology of the bay is unique. So we just put another tool in our toolbox for those surveys. And we've used drone imagery. We're collecting imagery of certain areas, those hard to reach areas that it makes monitoring difficult. And I'm not an expert on this. I don't have a lot of knowledge, but I believe that they're putting all of these images into software to try to get some algorithm to identify plants. So that is pretty cool technology. I'm not sure where they are with that, but we're in the process of doing that. That's another team I'm not really that involved with, but it pops up in time to time in the meeting, so I'm interested to hear where they're at. So Water Soldier monitoring results. We did find Water Soldier in 3 of the areas where eDNA kind of guided us to. So here in Muscogee Bay, we had eDNA hits there where we came up and went and did the survey and found Water Soldier. This area is known as Big Island. We had eDNA hits there, and we went and surveyed and found water soldier. And over here near Belleville and Trenton, we had hits, and that we investigated there and found some water soldier there. So eDNA has given us the opportunity to put a little less work in and kind of direct our efforts where we think we're going to find it through the eDNA results. So the achievements for the Bay of Quinte for 2023, through the eDNA positive detections, we surveyed 11 areas and overlaid the grid. We've implemented the drone work and the underwater camera. We've done an extensive shoreline investigation where the Bay of Quinte flows into Lake Ontario proper. The Invasive Species Center and the Invading Species Awareness Program did that over a 2-week period and came up with no plants. We have installed 45 signs around the Bay of Quinte for public outreach and education. The more eyes on the water that can help us identify where we're going to find this plant is crucial. It's important. We get a lot of phone calls from the public telling us where they've seen the plant, and it helps us, map it out a lot easier. And we've taken 141 eDNA samples, which helps us locate the plant as well. So that's the Bay of Quinte. So we're going to do a couple quick case studies about water soldier in other places in Ontario. So the Black River is a tributary to one of our largest inland lakes in Ontario, Lake Simcoe. In 2015, a provincial biologist found a major colony of water soldier when he was doing some work on the river. The MNRF, Aurora District, contacted the working group to consult and see how they would want to approach this population. And the decision to take an aggressive angle to eradicate was made because of the lessons learned on the Trent Severn Waterway. Without management, it will spread and grow. So a rapid response was implemented, and manual removal efforts and herbicide treatments were done in both 2015 and 2016.
Pardon me, I'm just going to interrupt for one moment, let folks know that quiz number 3 is up.
No problem. So in 2017, we monitored once a month from May to October, and no new water soldier was discovered, no new plants. So we implemented eDNA collection in 9 locations downstream from the main population that was present, wasn't anymore. And we were getting positive hits. In 2018, we continued to do traditional monitoring and taking the eDNA samples, and we were still getting positive hits from the eDNA samples. And we're scratching our head. We're going out and doing all this traditional monitoring, not finding any plants, but we also don't know how long these water soldier plants could be shedding material, even though they're not alive or viable. And again, this is a river, so it's flowing water, so it could be spreading across a larger area. So 2019, we were monitoring, but we were putting special emphasis where the plant was previously found. No plants found from 2019 to 2023. We continued eDNA collection, and we have had no eDNA detections of water soldier in the Black River since 2021. This is great news. We'll be going out in 2024 season to do traditional monitoring and take more eDNA. I don't want to say it, but we may be looking at eradication on this population, and it's due to that rapid response. Before we start, I started kind of with this rapid response. To me, I never thought it would be lasting almost 9 years now, but it's been taking a while. It's changed the way I view rapid response. I think those initial efforts really paid off in the long run. And it was a collaborative effort between provincial, non-government organizations. And I hope this one can be a feather in our cap to demonstrate that we can control this and eradicate this plant. So Red Horse Lake. Red Horse Lake is a part of a chain of lakes of the Gananoque watershed. It is located just north of Kingston in Ontario, and it is a lake beside Charleston Lake Provincial Park, which is a very popular provincial park here in Ontario. In September of 2020, we got a phone call to our invading species hotline from a concerned resident of the lake who owns a cottage resort there, saying that he thinks he found some water soldier. Myself and my crew went out to investigate, and indeed, we found a fairly large population of water soldier, really far away from any other populations that we've been kind of addressing. So it was a little concerning. So in 2021, we conducted surveillance and we did manual removal of satellite populations that were adjacent to one large population. And we performed a herbicide treatment in October of that year. In 2021, we went back, and again, the population looked like the reward had taken a bit of a toll on it. It wasn't as dense, but we still had those scattered populations, you know, plants breaking off the main population and settling out other places. So we cleaned those up and we conducted another herbicide treatment in October. In 2022, we went back, but this time we implemented the water soldier monitoring protocol. So we gridded the lake with the 50 by 50 meter grid with unique point intercept points. And we didn't find many plants that were across the greater landscape. Again, those little satellite populations adjacent to the larger population. So we started to implement the eDNA collection upstream and downstream of the known populations. And we did the manual removal of the small populations and performed another herbicide treatment. And we're seeing this population just shrink. But in 2023, we went back, we did our monitoring, and that main population wasn't there anymore. And I was kind of happy at first until we started doing our monitoring. And we found 13 small populations scattered upstream of the original population. And you can see in the map here on the right, right here at number 3 was the original population. And although that big population just broke up and settled out in all these little back bays. Now this lake is much different than Trent Severn Waterway. It's more of an oligotrophic lake. It has a very small littoral zone. It's deep. It has sharp drop-offs. So there's not too many places for this plant to fall out. So herbicide treatment wasn't really an option this year. The populations were maybe 1 meter by 2 meters, 2 meters by 2 meters. And we wouldn't really see the efficacy that we wanted using herbicide. So we manually removed 10 of the populations in September and were unable to find 3 of them when we went back. And this is a deeper lake, a little harder to see. So come October, we get a phone call to the Invading Species Awareness Hotline. A resident on the lake has found one of the populations right in front of her cottage. Invading Species Awareness Program, along with the Invasive Species Center, went up and we manually removed that population. And of course, I had to start poking my nose around. So we went to start to check on how well we removed the other populations in September. And lo and behold, we find 4 new populations. So given the time of year it was about mid-October, we really didn't have time to coordinate removal efforts. You got understanding that Red Horse Lake is about a 4-hour drive from our office. So we hired a herbicide applicator and treated with herbicide in November. A bit late in the year, but we're hopeful that it will. It's better than just not addressing it at all. That's what our rationale was. So hopefully when we go back in 2024, we can see the results that we want. We have a plan where we have a crew that would be housed out there from May until September this year. So there'll be a lot more time on the water, a lot more time for these investigations, and a lot more time to do surveillance on the adjacent lakes, seeing that this is one lake part of a greater watershed where there is attached water bodies with flowing water. And private ponds in Ontario. So since 2015, the working group has been addressing private ponds. As I said earlier, prior to the Invasive Species Act being implemented in 2015, Water Soldier was sold at nurseries and aquarium stores for who knows how long. So it was being sold to the public. I've even heard some stories where if you bought fish, you would get a free Water Soldier plant because of its nature of reproduction. So this is pretty wide area here. So the green dots are where we've eradicated it from ponds. We're sitting at about 75% eradication rate in these ponds. The yellow icons are people that we can't get in touch with. They've either sold their house and they don't live there anymore, so we don't have a point of contact for them. And the red dots, this red dot in particular, was made known to us by the landowner late this summer. And we're planning on going there in the spring and assessing the population and treating it with, well, either reward or Procelicor. This pond here, not really a pond. It is on somebody's property, and he uses it as a pond, but it is attached to the main Trent Severn waterway. So again, not a shocker that it's there, but it's one of those persistent ones that I think we want to address. It could be a source population feeding into the main Trent Severn waterway. So we did treat it with Procelicor. But it's a really unique pond. It's very difficult to access, and there's water flowing in and out. So I'm hoping we can revisit this one and try to eradicate that one as well. So on the horizon, where do we go from here? We're currently renewing the integrated management plan to rethink of how we're addressing water soldier populations in Ontario now for the next 5 years, especially with being in the Bay of Quinte, Lake Ontario now. We've had very successful management in the Trent Severn waterway, but our goal of eradication is elusive. We have a few hurdles to jump in terms of that persistent map I showed you. With the expansion to the Bay of Quinte, it brings a lot of new challenges. It's a vast area. The life history of this water soldier in this controlled canal versus large open water is going to bring some challenges and some lessons. Bringing on new stakeholders is vitally important, especially with such a vast area and limited resources. And we need to, we need to expand our partnerships and source new funding options. And that kind of is, I think, is every manager's has this issue. We have such a vast area we're covering, lots of water soldier, but those resources are limited. So that's where we're looking to go, and we're hopeful that we're going to get this under control, but it's going to take a lot of work. So that's kind of an update or what's been happening with Water Soldier in Ontario. And for more information, you can contact me. I'm Robert McGowan. There's my email. And these are members of the working group: the City of Belleville, Canadian Wildlife Services, Invasive Species Center, Lower Trent Conservation, MNRF, Ministry of Environment, Conservation and Parks, Mohawks of Bay of Quinte, OFAH, Parks Canada, Quinte Conservation, the City of Quinte West, and Trent University. If you're interested in getting involved in the project, reach out and we can have a conversation. And thanks for listening.
Thanks, Robert. So I'm checking the, let's go back to, sorry, let me go back to our questions. We've got our questions on. So show the screen. There we go. I'm just checking the chat and the questions box to see if anybody's typed anything in. Not seeing anything in questions. Let's see about chat. Well, while folks are gathering their thoughts, I'm going to ask some questions. So I'm curious, how does one do hand removal of something so sharp? I'm just curious, like what kind of equipment do you need to do that?
Well, we modify a pool pole with a thatching rig that we attach to the bottom.
Okay.
Usually long sleeves, pants, and gloves do the trick. It's not razor sharp, but
Okay.
you know, it'll break your skin. But usually just taking some, you know, wearing proper clothing and some PPE, some gloves, you don't really have an issue.
Okay. And I noticed that you also treat the plants with herbicide fairly late in the season. Is there a reason for that?
Yeah, so the rationale is a lot of it's cottage country. So a lot of cottagers are there. And with Water Soldier being an evergreen, we don't have as many run-ins with the public. They're not using the waterway like they would in July and August and September. They've shut their cottage down and gone back to the city. Water Soldier is an evergreen, so it's still actively photosynthesizing in the fall. So we're able to utilize that time to get all the monitoring done during the summer and plan the herbicide applicators to come. We hire a herbicide applicator out of the States. So it's a process with crossing borders and all of that. There's a lot of paperwork, so that takes time. So we're hoping that some of the applicators in Ontario, I know that some are building capacity to address an infestation this size. Herbicide in Ontario is not like herbicide use in Ontario is not like herbicide use in the States. We have very limited tools in our toolbox. So we have Reward diquat dibromide, which has been around for a long time. So that's one product we can spray over water. Recently, we just had Procelicor approved to be used through Health Canada, but it's very new. We did spray Procelicor last year in the Bay of Quinte. We treated 2 hectares. We're anticipating good results from that. And we have used Procelicor in private ponds, and we've seen great results with that. So we're hopeful that we have another tool in our toolbox, but we are limited in the amount of product that we have to use.
Okay. So there's a question in the question box from Stephen Pearson. How well does Water Soldier disperse across deep open water, and how about in flowing water?
So in flowing water, it's a floating plant, so it's very mobile. Deep water, we see 99% of Water Soldier in no deeper than 2 meters. So 2 meters to the shoreline, that it loves it there, it thrives. We have found a few populations at 5 meters, but they really don't do much at that depth. I don't know if they're not getting the proper light. They don't seem to be too mobile. Really, with it being in the Bay of Quinte now, Lake Ontario, these are the questions that we've asked ourselves. How is it going to act in this open water, deeper water environment? So right now, we were bit on our heels when we found it, and I think we're getting a little more stable now to kind of investigate those questions and start taking a look in areas where we wouldn't traditionally find it, in those deeper water areas.
Okay, we've got some more questions for you. So we've got, let's see if I can open this up. From Todd Tram, is there any evidence of water soldier moving south to the States?
Well, I don't know of any evidence, but we can see that it's flowing out of the Trent Severn Waterway into the Bay of Quinte. And, you know, Lake Ontario is a binational waterway, right? So we have Lake Ontario. We've been surveying down in that area, but we haven't found it. Keep your eyes open, New York State. And the Gananoque watershed runs into the St. Lawrence River as well. So there's two potential avenues for it to get into US waters.
Okay. And give me a moment, I'm reviewing more questions for you. Let's see. Having trouble opening this box. Hold on a moment, sorry. All right, I'll do my best. Okay, so we've got a question from, Summer has released quiz number 4. It's for folks to let you know. Question for Ryan. When might your reference genome be publicly available? And this is asked by Alex Satola.
Hi, Alex. You can just kind of, it's not publicly available, not for anything, not for any reason, to try to keep it secret, just that it's a very complex genome, so it's allohexaploid. There's a lot of heterozygosity, and we're like constantly refining it. So one of the challenges that we actually have with the kind of QTL stuff that we've been doing is like constantly rerunning analysis because we modify the genome. So at some point we'll publish it as part of a paper, and I'm not exactly sure when that's going to be, but it's not like it's impossible to, nobody owns it but us, for example. So I'm sure we'd be happy to share it in collaboration or whatnot. So just reach out to me. I think we've been on email probably a few times, so it might be a good idea. Just pick up the phone one day and we can call and see, what you need and how we can help.
Thanks, Ryan. There's another question for you from Peter Grundy. Is crossing milfoils for susceptibility an option?
Crossing them for susceptibility, like trying to kind of drive susceptibility through a population? Is that maybe a little more clarification on the question? I might be able to answer a little better.
Okay. So while he's typing, I'm going to go to the next question that he asked. Can we predict where a milfoil, let's see, hold on. Can we predict where milfoil product resistance may be more likely?
These are good questions. I'm not sure if I'm interpreting the question correct or not, but I would say, like, generally speaking, like herbicides that have a very specific target site and a very specific site of action, those are probably more predictable for target site mutations, right? So basically, like the narrower the target is, the more likely, the more restricted the number of mutations can be that can lead to that resistance. The kind of insidious thing about it is that there's a lot of options for non-target site resistance too. So like metabolizing herbicides, sequestering herbicides, and that seems to be, from my understanding of kind of the synthesis of the literature, those are pretty unpredictable. Like the same detoxification or metabolism that may work on even herbicides that have different modes of action and different sites of action. So I don't know that that's a great answer. I think we probably have more of a synthetic understanding for target site resistance And the answer to that is going to be like when the target site is more specific, then the number of possible mutations that can lead to resistance are going to be fewer.
Okay. Next question we have is from Mark Heilman. Hi, Rob. Thanks for your presentation. In terms of phenology, what is the annual germination rate of turions?
I don't have that information, Mark. I know there was some work being done through Trent University about turions. I think it's Robert Canning's paper. I can shoot you the paper. I have it on my desktop. I can email it to you right after this session if you want.
Okay, another question for Mark. Do the majority or almost all germinate in the late spring? Let's see, or early summer, or is there a good fraction of year-to-year carryover?
I know that we've seen them in the spring after they just it's almost like popcorn, they sprout. So they do overwinter. I don't know the exact answer on that one. These are good questions, but I'm not sure if that would be in the Roberts paper or not. I don't remember reading that. So I think the majority of the reproduction is done through the offsets. There were studies done with Eric Sager, who was Roberts' professor, where they put the turions in cages at the bottom of the lake, and I'm pretty sure it was inconclusive. I think they were doing that at certain depth stratas and everything. There wasn't a clear answer on that.
Okay, so Mark is saying, to clarify, is there documentation that turions remain dormant for more than one winter?
No.
Okay.
Not that I know of.
Okay. So then let's see. So going back to Peter Grundy's question, we've got, yes, can we drive susceptibility or temper resistance where it is occurring? Is what he was asking about earlier regarding the milfoil.
Yeah, it's a good question. I think, yes, it would be theoretically possible to drive susceptibility to a population. It's just a little different than maybe like if you're familiar with kind of like mosquito examples. So because these plants are both clonal and sexual, probably some sort of way of driving a gene that you want in a population through clonal reproduction is probably like in my mind, the more likely possibility, because within a lake or within a water body, the vast majority of biomass is produced asexually. And so in terms of kind of like using sort of like gene drive technologies that you might be aware of, I do think it probably would look a little different or a lot different for these clonally reproducing organisms. The other potential way, but it is through trade-offs. So if there's a cost to resistance, then, I mean, sort of the intuition is if there's a cost to resistance, like a growth cost, then by not using the herbicides or rotating herbicides and sort of going on these kind of, herbicide holidays, if you will, you maybe could remove resistance that way, but that depends on there being a cost, an appreciable cost to resistance. And I don't think we have a very good understanding of that at all. The one thing, in the state of Michigan, we have one strain that's really a fluoride resistant strain that's really, really common. And so, it obviously does just fine or fine enough in the absence of herbicide. So, yeah, that's my answer, I guess, to that question.
Okay. I believe we have another question. Let's see. Thanks, Ryan. We'll definitely reach out at some point. Would be happy to chat. Another quick question. When you made your mapping population, you only used progeny F2s, I assume, that exhibited the extreme response. Are you at all worried that you may miss any genomic regions that may be in linkage that could rescue some of those progeny, i.e. perhaps a progeny would have responded poorly, been at an extreme, but was rescued by another genomic region in the linkage?
Yeah, so we've for the one population we've done F1s and F2s. So like to map them, it just matters that the population is segregating. So I think typical, sort of our typical genetic mapping between homozygous lines, you need to have F2s to get segregation. So we have segregation in the first generation. So we did a QTL analysis on the F1s. We carried those to F2s and have done that as well. In terms of like the bulking, I mean, we certainly can miss, like, I mean, we're kind of biased towards finding genes of larger effect in doing it that way. So do I worry about it? Yeah, I worry about a lot of things. I'm kind of prone to worry anyway. So yeah, I mean, we certainly can miss things. And in one of the slides I had, like, one of the things we're kind of trying to dial in a little better is like the bulking logistics. And so there's this trade-off between, given a number of plants in the mapping population, there's a trade-off between the number of individuals that you have for your sample size and like how far in you're going into the middle of that distribution. So like ideally you'd have lots and lots of plants so that you can choose the most extreme bulks and still have good sample sizes for these permutations to compare the susceptible and resistant. And so that's one of the things I really worry about with our mapping populations. Our mapping populations aren't really all that big. So I guess that's a very long way of saying, yeah, I worry about it. But you know, what we're hoping to do is find markers that have some, that explain some good fraction of phenotypic variation. We're sort of keeping our fingers crossed that we have these like genes of major effect. And then, if we can screen populations for that, then I think it would still be better than doing what we do now, which is essentially qualitative at best, if anything at all, for sort of gauging resistance or susceptibility. And so our ability to predict it is essentially zero on any new strain.
Okay, and Alex also notes, and thus exhibited an intermediate response. Okay, I'm not seeing any other questions. No, I have one. I'll just throw it in there for Ryan. So have you thought about expanding your sample distribution to other areas of the US? I know you seem to have focused on certain states. So is there any ideas about expanding that, or you're thinking this is enough for now?
Oh yeah, I mean, so right now it's whoever wants to send us stuff. So we are biased towards the Great Lakes states. In part, that's because of the history of where the research started, which is when I was in Michigan, and then also because those states are very, very lake rich, right? It's probably 50,000 lakes between Minnesota, Michigan, and Wisconsin. We do have samples from around, and at the moment we're not like really targeting any particular areas. A few years back we had some GLRI funding, and so we really tried to kind of spread out across the whole, the entire Great Lakes basin. So we certainly welcome samples from anybody, anywhere. Yeah, and it'd be great to really capture the, I mean, we'd like to have every lake that has water milfoil in milfoil map.
Okay. Okay, so I'm told that if I scroll up, there's a question from Jack Witness. Let's see. Sorry, it's in the chat box. Give me a moment. Not seeing that, sorry. Go back. I seem to have missed one. Hold on. Okay. Okay. Do single lakes frequently have more than a single strain of Eurasian water milfoil? If so, please discuss treatment considerations.
That's a great question. So we most commonly find a single strain. So and I should put that in some context, we typically shoot for sampling 20 to 50 plants. And so our most common result is a single strain. But that said, there are many, numerically many lakes that have 2 or more strains in them. When there are multiple strains, what I would usually recommend is like following strain proportions over time. So if you do an herbicide treatment and let's say you get 80% control, right? But there was 2 strains in there, you wouldn't necessarily know, okay, did you kill 80% of each of them? Or did you kill one at 100% and one at much less than that, leaving you that 20% over, right? So the answer to that question would be really informative. And so usually I recommend that people, if they can, and I would talk to me about the details of doing it before doing it, because there'd probably be some situations when I would say it's probably not worth it to do it. Don't waste your time or money. But following sort of monitoring strain composition over time can potentially be informative for something like that, to kind of see if any strain volunteers itself as being disproportionately not controlled by a particular herbicide treatment.
Okay, thank you. Any more questions for folks? Not seeing any more in the question box. Okay, then I will move. Let's see if I can get this to shift down. Not so much. Okay. There we go. Okay, so I wanted to say thank you to Ryan and Robert for presenting today. It's been really informative. And I also wanted to let folks know that they could stay tuned for more offerings this year from NEAPMS, and a recording of this webinar will be posted at www.naapms.org. Thanks everybody, and have a great rest of your afternoon.
Thank you.
Thank you.